Yeah, today I'm going to take you a little bit on a journey of how we can measure the human brain structure and function in living humans in order to understand healthy brain organization, a little bit of development, and a little bit of disease. I want to start this obviously by saying that much of this work would not have been possible without the friendship and intellectual input of my friend Jonathan Smallwood, who tragically passed away very recently. So many of the concepts that I'll discuss, but like a lot of also the fun and international collaboration, it's really also thanks to him. And I think many people here in this room have really been influenced by how he helped us to kind of think about the brain and about structure-function relationships in human brain. So my objectives for today is that I'm going to just show you like a little bit of the tools and toys that we have now to try to measure the human brain in vivo. I know this is a big brain meeting, but I'm moving a little bit away from histopathology. You get like a little bit of histological validations here and there, but it's like not the main focus. I'm going to show you a couple of open science data tools, mainly from our group, to process the data, to co-register data, to bring data into the same space. And then I'm going to show you some applications for healthy brain organization, but also to bring things to the clinics. So, I mean, as neuroscientists, what are we trying to do? I think we're trying to understand how the brain is generally organized. We're obviously trying to, and this means we want to see how something that has a complex network structure gives rise to cognition and function. We also want to understand beyond these generalized principles in what makes every human unique. So we try to understand individual variations, and then we also want to help patients who suffer from prevalent conditions of brain function. So this is always like one of the starting slides for this. So in the olden days, this was usually done by trying to derive a cortical map. And as you are all very familiar with, there's like many different ways with which we can get cortical maps. Initially, this was always based on foundational post-mortem work. So myeloarchitectonic atlases, cytoarchitectonic atlases, and here the premise is really that areas that have different microstructure, obviously also they harbor different functions, and they also have different connectivity profiles. And then in parallel, folks like Walter Penfield, so this is the fella who founded the institute where I'm working in, they have done intracranial invasive experiments, both recording but also stimulation, in order to localize different brain function in cortical areas. And obviously here then the task was a little bit to map between these two things. So to try to kind of bring things that we understand from lesion experiments, that we understand from stimulation studies, to these databases that are coming from different individuals. So you see where I'm going, that this is usually like a leap of faith, that we're trying to map from one data set to the other, so we don't really interrogate structure and function in the same individual. And Jessica, who's, I think, over there, like in the back, have you given your talk today already, or is it tomorrow? It's tomorrow. So tomorrow you hear like a lot of insight about this, that there is potentially like at the level of imaging, but also when we want to use reference data sets, there's potential challenges at least that we have to address, because there might be inter-subject registration problems. There are some limitations on the data sets that we use. As you know, like some of these really foundational data sets like BigBrain, they're extremely complicated to generate, so we don't have so many of them. So we make inferences coming from small sets of, however, very densely phenotyped subjects. And then this is like also something that's coming more from the MRI world or from other studies, is, as you may be very well familiar with, is that there's like a lot of inter-individual variability in like these more higher order association areas. So we don't really find a common ground with few subjects, or at least it's like an ongoing problem. And how can we find correspondence between data sets and between people? But tomorrow you'll hear like a much better explanation of this. So with MRI, which is my main tool, we have abilities to interrogate structure and function in the same individual in the same session. My analogy is always the Thermomix, which the German chefs here in the room may know. So this is like a machine, and you can load a couple of different programs on it, and then it cooks you some recipes. So in the MRI, the only limiting factor really is time and subject patience, in a way, that you can run a structural sequence, you can run microstructurally sensitive sequences, diffusion, ASL, a lot of things, and importantly also functional sequences where you can try to look at hemodynamic evolution over time and then kind of try to interrogate different functional networks, such as the default mode network. You can also decompose resting state fMRI patterns into large-scale networks, and this helps you to really map functional networks as well as microstructural organization in the same individual, in the same session, and also at the group level. Alan is in the room, and he's one of the inventors of the MNI space. And this is a stereotaxic space that has been developed to bring different individuals into like a corresponding coordinate system. And in a way, it has kind of paved the way for neuroinformatics for us to map not only different MRI contrasts into like a single space, but also to combine MRI with other different neural features. And these are like just a shameless pitch on like some of the tools that have also been evolved out of the HIBALL initiative. So we are really thankful for like all the people who have been working on this. So we have BigBrain Workshop from Casey, who's also in the room, trying to bring MRI data in the same space as BigBrain space. Can the contrast be changed a little bit maybe? Can I do this? Or just the lights out? Thank you. Okay, that's better, hopefully. So, like BigBrain Workshop, but we also have a couple of other toolboxes in which we can, for example, co-register different MRI modalities into the same space. HIBALL maps and HIBUnfold. Jordan is also in the room here, also like one of the HIBALL trainees who has done like really fantastic work in trying to bring together different neuroimaging contrasts, but also imaging, big brain, and other data sets into the same reference space. And this is just like a very small example of multimodal MRI projects. Alain talked a lot about big data, so I'm not going to talk so much about this, but we can obviously now also use data sets like ABCD and UK Biobank to really make population-level inferences in a lot of subjects that have been scanned the same way. But we can also now increasingly use so-called precision MRI data sets where just small groups of individuals, but hopefully growing groups of individuals, underwent extensive scanning. So here, this is the Midnight Scan Club, which you may know. I think it's 10 individuals, and they have each undergone like a couple of hours of scanning. It's called Midnight Scan Clubs because the scans were done after hours when like the other protocols were not running. Why does this matter? It's not only to characterize, population level variation and to maybe more deeply characterize brain functional network organization in single individuals, but most of these MRI sequences, we can actually also translate them readily to the clinic. Here's one example of an initiative that we are leading in Canada right now, where we're trying to really define epilepsy biomarkers in both children, but also in adults with epilepsy. And we have been harmonizing all the sequences. So we're standardizing the way that images are acquired. We're standardizing the way that images are being analyzed and hopefully processed and as well as analyzed in order to really find biomarkers for some conditions that might not be so prevalent. I mean, epilepsy is a prevalent condition affecting about 1% of the population, but specific subgroups with very specific lesional entities, they're still quite rare. So we need to really aggregate data that is of high precision. And at least one idea could also be that when data has been acquired with very high precision, with very high quality, you might not need as many training examples in order to identify really powerful biomarkers. One thing that I'm extremely excited about is 7 Tesla MRI. So some sites now increasingly install these systems. So this is higher field strength than the commonly used 3 Tesla MRI. And here you can basically, hopefully appreciate by how good we are getting with in vivo imaging when resolving brain microstructure. So this can be done in 10 minutes at a 7T. So this 500 micron isotropic voxels. And if you do more than one of those, so say you have a bit more time to spare, you can even upsample these things intelligently to get like 250 micron in vivo imaging for like patients, but also for healthy individuals, to characterize cortical lamination patterns, to characterize maybe even atypical lamination, which I'll come to this a little bit later. In like some epilepsy syndromes, it's really the cause of the seizures. This is just to show like the gap from 3T to 7T MRI. So here you can really see how we can boost the resolution. So these images basically take the same time to acquire. So it's really exciting, I find. And when the system was installed in our center, we were all a bit worried that, yeah, there's like a lot of inhomogeneities, it's more complicated to use. No, it's not. So I think the images, except for diffusion MRI, overall they're really exquisite, and it's ready for prime time in a way to really develop biomarkers. Problem is obviously the cost for these systems. So this is a data set that we've shared. Donna Gift-Caballo is the first author here, and where we've basically made 10 subjects available who have undergone these 3 repeated scans. So everyone has been scanned for 4.5 hours. And we have the microstructurally sensitive sequences. Some of you may know these, MT, QT1, so you can map myeloarchitecture in vivo. But then we also have obviously resting state fMRI and a couple of tasks. So in this cohort, we initially released 10 individuals. Here's the QR code if you want to download it while I speak. But by now we have like more than 30, I think, who underwent like these sessions. So it's quite practical. I also want to point your attention to an initiative that's currently going on at the Neuro. So this is led by Julien Doyen, and this is a lifespan 7T MRI data acquisition initiative using similar sequences than the ones that I've just presented. And here 600 subjects will be scanned from 6 to 80, I think, and using really high definition 3T, 7T MRI. And I mentioned already the initiative that we are currently leading for epilepsy data exchange, but we also want to make this together with other sister 7T sites, for example, Western. And if you have epilepsy patients and if you have a 7T scanner, shoot us an email. I mentioned the M&I space already as like something where MRI actually has led the way that we standardize our methods and our concept of thinking about the brain. Here's another one that has become like really powerful in order to standardize the way we name files. So it sounds like almost a little bit low key, but it's really important because across different initiatives before, different investigators, they had different names for different sequences. So now there's really like a standardized way to name files, both at the raw file level, but also how to name derivatives. So for people who do software engineering and who develop apps, this is really the missing piece in a way, because we can now assume that data are available in bits, and this has been very successful for MRI data, but also been adopted now for other modalities. Actually, there's EEG bits, there's PET bits, MAG bits, I assume. There's even intracranial EEG bits. So a lot of different. There's a lot of success in like how we can try to organize our data and subsequently process it. So I mentioned the Mika pipe already. So this is like the tool developed by Raul Cruces. Works for 3T, 7T. It's a BITS app. So it should work out of the box for any BITS data set that you have. And logistically, it's a little bit of a nightmare in a way, because it has like so many different components, and you really have to make sure that they work together. However, we have functioning Dockers. And at least in the lab and in like some sister sites, they also work relatively well. So, but it's obviously they're complicated to make them work on like every different data sets, but now with like increasingly users doing the testing for us and running it for us, we also can find more bugs. And I think this is like really the beauty of open science, that many people use it, they let us know what doesn't work, and we're trying to make updates for it. So hopefully this is already working with the current version, and there's like a new version coming that has like even stronger co-registration abilities. Jordan has developed a really successful tool, HIBALL, during his time in Western, and like now he's continuing to develop it to make it even better than it was before. So you can get this here, and then recently Jordan used the unfolded way that we can deal with hippocampal subregional organization to kind of map not only different subjects into the same coordinate system, but also to map different data sets into the same coordinate system, and this has recently been published in Nature Methods. And this is really like a data warehouse where when you do a hippocampus analysis, the way to do it basically is first you run HIBunfold on your data. This works on conventional data, but also 70 data, works on a variety of different contrasts, and then you basically have like all these different maps at your disposal, so you can compare the patterns that you find, be it from your resting state fMRI study or be it from your task-based fMRI experiment, with like these different maps. A little bit like the neuromaps that Bratislav has pioneered for the neocortex. So but here just in a very hippocampus-centric fashion. And we're currently working in a top secret project on hippogenes, so stay tuned. So this I'll present next time. When we have all these different data sets in the same reference space, so structure, microstructure, function, obviously we're often faced with like high data dimensionality. So sometimes also with connectivity. You can imagine that when you do a resting state connectivity experiment, you might have connectivity maps for each of these different regions. So you have a matrix basically, and how do you explain the matrix maybe to surgeons or to clinical end users? And one way to do this is to basically explore different techniques for dimensionality reduction. And here's like a tool that you can use for this. It's called BrainSpace. Reinder Fosterval developed this during his PhD. It's basically doing PCA and nonlinear variants of PCA to map spatial variation in specific interregional features back on the brain. And you're possibly all very familiar with like these sensory to transmodal gradient that you can get from this, but you can also get other gradients. So, and here we can get this, for example, from microstructurally sensitive data, from functional connectivity data. Another tool that we can use, this is BrainStat. So when you have your data on the surface, be it from the different contrasts, or be it from the gradients themselves, you can then obviously also use between group comparisons. You can compare a single subject to a normative data set. So currently, this is like more geared towards group level analysis, but we're currently working on an addition of this to also incorporate normative modeling in order to map really patient-specific deviations relative to a large baseline population, which can sometimes really matter for individualized patient diagnostics. Here you can see one of these applications already to resting state fMRI derivatives in a multi-center cohort with temporal lobe epilepsy. So this was led by Kichi from the lab. So we had about 300 TLE patients, 300 healthy controls across a couple of sites. And then from the resting state fMRI, we didn't really look at the connectivity per se, but we derived derivative features that are either sensitive to like these more local signal variability features, to more regional, but very local regional connectivity. So we computed ReHo here. And then we also had a node strength measure that's more of a graph theoretical measure of overall connectivity degree. And then for every patient that we had, we placed them into an age and sex controlled population. And then we basically quantified W scores, which is an age and sex corrected Z-score. So this is like the simplest normative model that you can build. And when you do this, obviously, 2 minutes? I might need a little more. What Kichi then did is he could not only show proportions of deviations relative to controls in patients, but also relate these features to different clinical measures, including surgical outcome. So you're going to possibly hear about like this beautiful work from Casey also a little bit later. So this is really groundbreaking work where we have tried to validate some of these gradients between imaging and histology. The papers are here. You can also talk to Casey in a bit. I'm going to skip over this because I think maybe you'll talk about this a little bit later. Yeah, she can. For sure you can. And basically showing a little bit the correspondence of gradients that we can get from in vivo MRI as well as from histology, and then also making the bridge to mapping these to functions. So here now we can look at the associations between microstructural and functional gradients at the group level, but also in single subjects. So we can do direct structure function studies in large-scale cortical systems. We can also do this in specific systems, such as the default mode network. Here again, resolving fine-grained microstructural organization, both based on BigBrain, but also using in vivo MRI data sets and then relating patterns of microstructural variations to default mode connectivity, but also to default mode signal flow. We can look at this with development, and then finally, we can look at this in atypical development, and this is maybe like what I wanted to talk about primarily today. There is a specific, very epileptogenic entity. It's called focal cortical dysplasia. Some of you may know this. So this is a very common cause of drug-resistant epilepsy, very prevalent in children, and very difficult to find on MRI, and even on histopathology, sometimes it's difficult to see. So usually the FCD spectrum is characterized by some degrees of cortical dyslamination. So all the FCDs, they have atypical cortical layering. And then in like some of the subtypes, like FCD type 2B, you might also have cytological abnormalities, dysmorphic neurons, balloon cells. So consequences of mTOR dysfunction in the cells. And these pose really a challenge for patient-specific decision-making because these are developmental malformations of cortical development, but then the clinicians don't see them. Even expert radiologists may overlook them, so there's no way to do surgical planning with non-invasive techniques. So we are working on a research program where we're really trying to use 7T MRI, but also 3T MRI, together with feature engineering approaches, to really find these lesions better. Here you can see one of those. So this is in the motor cortex from a piano player. So obviously it's very challenging to accurately delineate these lesions. And yeah, you can see not only how you can maybe get higher sensitivity when going towards 70, but maybe also how you can increase specificity. So you might get less false positive findings because you might be confronted with less partial volume effect, and this could be very good when we design automated classifiers. Another thing that might be really helpful for the design of automated classifiers is biology-inspired feature engineering approaches. So this is work done by Seok-Jun Hong. He did a postdoc in the lab, like now already a decade ago, and where we basically derived cortical surface models on different contrasts. We mapped them using co-registration techniques into the different spaces, and then we tried to model FCD appearance based on computational neuroanatomical techniques. And this was obviously very helpful to differentiate between different FCD subtypes, but here you can see, for example, work from Ella Salas, that's now work coming from the lab, where we used these different features in specifically labeled FCD lesions, and we compared lesional alteration load using W-scoring approaches to the contralateral homolog region, but also to ipsilateral non-lesional cortex in order to really find sensitivity and specificity of these different FCD-related features. So there's cortical thickening, there's cortical intensity blurring, and there's also some microarchitecture-inspired features like the intracortical moments from cortical layering. We can do something very similar in temporal lobe epilepsy, which is another very common epilepsy condition, usually localized to the hippocampus and adjacent mesotemporal lobe structures. So here we can use similar unfolding techniques such as the ones that Jordan had developed, and then we can really map different features that are sensitive to different degrees of microstructural alterations in the temporal lobe. Just one more sec. So we can, for example, map atrophy, we can map T2 intensity increases, mean diffusivity, and this is usually always done based on preoperative MRI features, obviously, but we can then also validate what we see in vivo with histopathology. And indeed, when using the surface-based features in linear discriminant analysis, we can almost with perfect accuracy classify histopathology, and this becomes more and more relevant because in the epilepsy world now, there's like a shift towards non-invasive or towards minimally invasive approaches. So we might just not get access to tissue anymore because there might be more neuromodulation happening, there's more thermocoagulation, so there's really no access to tissue anymore for at least certain patient subgroups. So this I mentioned already. I think I can skip over this. So these are like the normative models that we can also use for function. And obviously, when we have very extensive neuroinformatics approaches and we map things to complicated etheric cortical surface models, the problem is like, how do we bring this back to the individual patient? So we can do individual patient analysis by using, for example, normative modeling, but we are currently also engaging in some approaches where we're basically trying to map things back to the clinicians. So this is funded by HIBALL project, where we get individualized patient reports, and they are used already, like in the seizure conferences in Montreal and in other centers. And then we have also ways to burn the data back onto DICOM. So we basically have a tool that goes from DICOM to surface back to DICOM. And this is then really amenable for neuronavigation platforms like Medtronic, BrainLabs, Inisha. So here you can see, for example, 2 different patients. Here you can see the deviation map. Here you can see an AI-based result. And the contrast doesn't show it, but this is basically resected tissue. So this is this retrospective validation of what we do. So it does not directly inform clinical decision making at this point, but we can overlay it on the images and we can just see the accuracy of this. We can also use similar approaches, obviously, to map the intervention. So to wrap up, I hope I have convinced you that there's like a lot of excitement with imaging, but also with high-resolution imaging, and 7T in particular. We stand on the shoulders of the giants, so a lot of things have been already developed, including standardized spaces, cross-modal registration, cross-subject registration, but also things like BITS. And in the epilepsy world, there's also standardized imaging sequences. I think this is like a good way to go, that we try to standardize things as much as we can. This then helps us to really get a precise picture of anatomy and function in vivo, and we can get this ready for clinical prime time. I had this also, like if ever you're interested in FCD, like this lesion that I talked about, come to the Neuro next year. I think you're up for a treat. We have some really good speakers talking about the genetics, about the microstructure of FCD, but also brain networks and imaging. And if you're looking for a job, and if you are looking for a job, we have like some we're looking for clinicians, clinician scientists, but also full-time researchers to join our epilepsy group. Thank you for your attention, and thanks to the support from HIBALL and all the colleagues and collaborations.